Okay, so welcome back, grade 12s. So this is going to be a little bit longer of a lecture. I tried to go through it. I've been trying to kind of eliminate pieces of information that you don't need to know. But when I went through this lecture, it was very, very difficult because a lot of it you need to know in order to be able to understand other parts. So this will be a little bit longer. I apologize. I'm going to give you kind of a shorter worksheet, but it is kind of crucial to understand mutations. Okay, so let's go through this. So what is a mutation? A mutation is an error in the DNA sequence during replication. So while DNA is replicating, that's when a mutation happens. Okay, you try and remake the exact same copy of DNA, but it doesn't form perfectly. So the errors occur during replication. Now, since the error is now part of the DNA, all cells that are created from this cell that contains the error will create a bad protein. So it's not just the error occurs in one cell, but then that one cell becomes two. So now you have two cells with the error. Then they replicate. Now you have four cells with the error. So once a cell gets an error, it just keeps replicating with that bad error. Now, if the replication error occurs in the testes or the ovaries, the error will be in your gametes or in your sperm and your eggs. And then this can be passed on to your offspring. So that's what we call genetic diseases or hereditary diseases. Okay, not all diseases are like that. Okay, you don't pass on all of your hereditary diseases in your sex cells. Okay. Now, if the error is in a somatic cell or a non-sex cell, the error will die with the individual. So if you develop, say, skin cancer later on, you can't pass that skin cancer back on to your offspring. Okay? Even if you have, say, skin cancer while you are pregnant, your baby is not going to have that skin cancer unless the skin cancer is broken apart and some of those cells have been transferred. Okay? All right. Now, are all mutations harmful? Usually we do think of them as harmful. When you think of a mutation, you usually think of it as a bad thing, but there's a lot of good thing, okay? Mutation is what drives us to turn from bacteria into humans, okay? That's what drove evolution. So mutations are not always harmful, okay? So no, some may be harmful or which can lead to developmental diseases like cystic fibrosis. Some are positive, okay? Then traits that improve our survival chances. Things like, evolving a mutation that creates a different protein for, stronger muscles or a bigger brain size, okay? That's an example in humans, is that we've had mutations that increased our brain size, which enabled us to be more complex. Okay? And then there's a lot of mutations that have no effect, okay? They're undetected, where the mutated gene may be masked by a normal gene, where you maybe got a mutated copy from your mom and then a regular copy from your dad, or maybe the mutation happened in a gene that's not essential, or maybe the mutation happened in an area that doesn't code for anything. Okay? So sometimes you can have a mutation and not really have the effect from it. Now there's two main types of mutations, okay, which we call point mutations and chromosomal. So point mutations, actually I'm going to talk about chromosomal mutations first, because that's where we kind of started with in grade 11. So in grade 11, what a chromosomal mutation means is that an entire chromosome gets mutated. Okay, so that, if you look at all these little black dots that are on the chromosomes, so let me just get a pen. So if you look at a chromosome, and I'm just going to kind of grow one up here. Okay, this is what the chromosome looks like, and there's all these kind of little black dots. These little black dots represent genes. Okay, so codes for them. So that's gene, that's a gene, that's a gene, it's a gene. So if you have an entire chromosome, okay, that has an error in it, like you have an entire error in that chromosome, you're going to have this gene with a problem, this gene with a problem, this gene, this gene. You're going to have lots of different proteins that are going to be off. So these are very usually severe mutations. Usually it's going to completely cause death. Okay, you usually can't survive a chromosomal mutation because you have way too many genes that have been mutated, way too many proteins that are off, and it usually always leads to a miscarriage or death. Okay? Now, there is the rare time that we can survive some chromosomal mutations, and that's what we talked about in grade 11. That's like things like Down syndrome, things like Klinefelter's. Okay? Now, even when we went through those categories, with the exception of Down syndrome, most cases the babies were severely disabled, and they were severely disabled and usually had early death even if they were able to survive, because they had major problems in a number of different genes and a number of different proteins. Down syndrome is an exception where they're quite capable, very capable individuals. It's kind of a miraculous that they are able to do so well with the number of mutations that they have. Okay? Now, what's less severe is what we call point mutations, because what point mutations means is it's not the entire chromosome Okay, so if I redraw my chromosome with my different genes, a point mutation means that one gene is done. The rest of the genes on that chromosome are completely fine, but only one gene is affected. Now, this still can be severe, like cancer, okay, one gene being affected. But it can be also other not so severe, like one gene affected where I lost the hair color, okay, where it knocks out, say, the instructions for how to make dark hair color. Okay, one gene. I'm not losing a whole bunch of different genes. Okay, so we're going to start with point mutations because we went through chromosomal mutations in grade 11, and we kind of focused on that because we were talking about chromosomes. So we're going to focus on more the point mutations in there, where it's like one gene that gets screwed up, and there's really three types. Okay, you can have a substitution, which you just swap out one letter for another letter, and then these two. These two are a little bit more severe, insertions and deletions, where you insert a letter or you remove a letter. Okay, the difference between terminology here. So when you substitute, think of a substitute teacher. Okay, it means that one teacher is gone, like me, and there's a new teacher there. We've replaced each other. Okay, an insertion would be like me still being there, and then I bring in a supply teacher, and there's two teachers there. Okay, or a deletion would be like me not showing up to work, and then they never bring in a supply. Okay, that's a deletion where you've deleted the teacher, but you never replaced them. So some terminology will help us understand what each of them mean. So the first one we're for is at substitutions. Okay? These are usually not that severe. Okay? And really what happens there is a letter has gotten swapped out. Okay? So basically you kind of look and you say, okay, there should be a T there, but the T got replaced by a G. Okay? Now it's not that I inserted a G, it's that I took that T out that was there and then put in a G. So it's substituting it. Okay? If you kind of look at it like a phrase, if we take this is the original phrase, the fat cat ate the wee rat, if those are all the words, it's like a substitution mutation is taking one letter, just that H, and changing it. But the rest of the sentence is perfectly fine. And you can still pretty much make sense of the sentence. You can probably look and say, oh, that was a typo. It should say cat. So usually not a huge effect. Okay. Now, what this normally results in is what we call a missense mutation. And that means that it just alters a small thing, and it's going to have a slightly different amino acid sequence. Okay? But not usually a huge thing. You're going to have like usually one or two amino acids that are going to be different. And I'll kind of show you an example coming up. Okay? Here's an example, sickle cell anemia. Okay? So this is a substitution. So if we look, here is the original DNA sequence. Okay, and if you go across, you see that there should be an AT here. Okay. So what happens is during the mutation, and if you notice, okay, the mutation that happened is right here. Okay, if you noticed, it was a T, and now it's an A. Okay, and that was an A, and now it's a T. So because these bases have switched, what that causes is now a different amino acid. It should have been glutamatic acid, okay, in the polypeptide or the protein. But now most of the amino acids like valine, histine, leucine, most of them are in perfect order, but I have one thing out of place. And having that one amino acid out of place creates the bad protein and turns your red blood cells from looking circular to looking like sickle cells. Okay? So that's an example. Now usually this is not lethal. Okay, people can usually survive this. And again, it's less severe. It's a point mutation. One letter changed the protein, made my blood cells different. Okay. Now again, this is called a missense mutation because only one amino acid is out. Have one amino acid out, you're going to have an early stop codon. Now, the other thing that can happen also is what we call a nonsense. And a nonsense mutation means that you're not. So what that means is that your protein is going to stop prematurely. You're not going to have all the amino acids. If a protein needs, say, 10 amino acids, you're only going to make 5 of them. Okay. So an example would be, say, the original phrase is the fat cat ate the wee rat. Okay. You stop halfway through. You don't get the entire amino acid sequence. So the protein, this is going to be a little bit more of an effect because now you don't even have the entire protein. It's not just one amino acid's out. This is like a whole bunch of amino acids you're missing. So this is usually lethal to the cell. It's usually a much bigger mutation. Okay. Now, the other thing that can also happen is maybe there's not going to be any effect at all. Maybe there's going to be a silent mutation. Okay, and there's the case where, say, if on the third letter there's a mutation, it should be U, and it gets switched, and the letter turns and it turns to C. Okay, well, there's no effect because CAU codes for histine, but also CAC does. So even if you get this mutation and your letter mutates from U and it turns into C, it has no effect on you because you're still going to have histine in the right spot. And this occurred in the gizmo that I requested you to do last week. Okay? So that third letter there is really not that important in comparison to the first two letters. The third letter is what we call the wobble position, or really kind of a backup letter, where if it mutates, it's not as serious if you mutate the first two letters. Okay, so just to summarize, okay, we have point mutations. So you're going to change one letter, okay, and you're going to substitute it out. Okay, so you're going to take one letter out and put a new letter in, and it can result in a number of different things, like one, a silent mutation, okay, where we get a letter and it has no effect. The amino acid sequence is still perfect. Okay, so that's the perfect amino acid sequence. We can have a missense where you have one amino acid. Let me pick a better letter here, or color. One amino acid that's off. The rest of them are good. Methionine, lysine, and phenine are all still good, but one amino acid is off. That's called a missense. And then my last one, a nonsense, means that you have an early stop codon. Okay, it could be here. It could have 2 amino acids in the stop. It could have 3 amino acids in the stop. It just means that you have an early stop codon. Okay. And those are all the types for substitutions. And again, those aren't usually as lethal as the next one we're going to talk about. The next ones we're going to talk about is, well, what if I don't even remove a letter? I just throw an extra letter in there. Or I just remove a letter and never replace it. Okay? And those are called insertions and deletions. So look what happens when I do an insertion. Okay? When I do an insertion, when I just insert an extra letter, then basically what it will do is it will push all the letters to the right. Okay? So the best way to look at this is look at our original sentence that we've kind of been going through. So if the original sentence was the fat cat ate the wee rat, okay? If I put an extra letter in there, okay, and notice that all my words have 3 letters, just like how DNA is expressed. If I throw an extra letter in there, an X, okay, it has a way bigger effect than a substitution. A substitution, okay, then most of the words are still exactly the same, and just one word is screwed up. But here, when you throw an extra letter in, now everything gets pushed down. So here was the old word ate, but it's been broken up. Here's the old word the, and it's been broken up. Here's the old word we that's been broken up. And rat's been broken up. So an insertion basically throws everything off because it pushes all the letters down, and now everything has the wrong 3-letter sequence. Okay? So that's what an insertion does, is it causes what we call a frame shift. Okay? It means that everything now gets moved down one. Now a deletion will do the exact same thing. If I delete a letter, the same kind of thing happens. So let's go back to our original sequence. Okay? The fat cat ate the wee rat. And if I eliminate the C, so if I went through and I just delete that C, I don't replace it with anything, okay? Because if I replaced it, it's called a substitution. But this is just a deletion. So I'm just going to eliminate that C. That C's gone, and then again, look at the effect. It screws up every single word downstream when you eliminate a letter, because everything gets shifted to the left. Okay. So again, when we have these insertions and deletions, okay, it can cause a missense where you're going to have a whole bunch of now amino acids all screwed up, or it can also, if the deletion happens, and it can also cause an early stop codon, okay, known as a nonsense mutation. But the big thing here is that it's going to have a major comparison to a substitution. Okay, so let's look at all these 3 together. Okay, so the first one we're going to look at. So this is the normal amino acid sequence. This is the amino acid sequence that we want. Okay, so there is our normal mRNA, okay, and that came from our normal DNA. Okay, now let's see if we have a mutation. I'm going to highlight where the mutation is. It's right here. Okay? We get a mutation in the DNA, which leads to a mutation in the mRNA. And now when the mRNA is red, instead of glycine, we're getting serine. Okay? And that's what we call a missense mutation. Okay, one amino acid or a couple amino acids are off. Now, the other thing that could happen is maybe you get a early stop codon, okay, known as a stop or a nonsense mutation. Okay, and that's where the letter here changed the mRNA, and so on. And then a frame shift. Okay, frame shift. What we basically did was we eliminated that A right here. We eliminated the A. Now everywhere downstream all has the wrong amino acid sequence. Okay. Now this can be a little bit tricky. As you go through this, okay, it should be a little bit challenging. All right. All right, so generally speaking, although results vary, the order of severity is the least severe is going to be a silent. Okay, the next is going to be a missense, and it depends. Okay, excuse me, on one amino acid off or 10 amino acids off. But if it's only 1 or 2 amino acids off, then it's usually less severe than a nonsense. Okay, nonsense means early stop codon, and it also depends on how early. Like if you have 100 amino acids and the stop codon happens after the first one. So now you only have 1 amino acid when you need 100 to make the protein, it's going to be really severe. But if you get 98 of the amino acids done and then the early stop codon gets initiated, then it's not going to be as severe. And by far, a frame shift is the most severe. Okay, and that's insertions or deletions, because now you're just going to screw up all the amino acids downstream. Okay, now we're going to roughly touch on chromosomal mutations. I'm not going to spend very much time on it because we did talk about it in grade 11, but I just kind of want to show you the difference between a chromosomal and a point mutation. So again, if we had a point mutation, that would be like me screwing up this. Let me pick a better letter here. Getting a mutation in just this area right here that I highlighted in black. That gene gets knocked out. But all these genes that you see in these dark green are all okay. Okay? Chromosomal mutation means all these genes, all of them got knocked out. Right? Pretty severe. Now, not all chromosomal mutations knock out everything. They do sometimes will only do a small break. So in this location, this example is called the translocation. It means only the bottom half of the chromosome gets broken. Now, again, I'm not going to spend very much time on this because it's something that I'm trying to cut. Okay, so I'm just going to kind of skip through these. Now, the causes of mutations. We do need to go through this. There's 2 different ways that you're going to get a mutation in your body. It can be spontaneous, and this is just an error during a replication. Okay, it doesn't have anything to do with the way that you're living, like your lifestyle choices. It has everything to do with your genetics and your ability to replicate pretty consistently, or your ability to replicate not so consistently. Okay, and some of us are just born to be better replicators than others, okay? And those who don't replicate very well are prone to mutations, okay? So that's just spontaneous. This is genetic, okay? You have no control over this. You were born with it. You shouldn't really worry about it, okay? I know it's hard not to worry about it, but you have no control. You can't change it, so you're just going to have to forget it. This is what you need to worry about, the environment, okay? The bottom part, right? This is called induced mutations. And these are errors caused by the lifestyle choices that you've done. So it means that you now have a mutation because you were exposed to UV radiation or X-rays or pesticides or cigarette smoke. Okay, you basically induced an error. Okay, this wasn't a genetic thing. This was the lifestyle choices that you've chosen have caused you to start changing your DNA. Okay. So there's a few main ones, and we're only going to be talking about induced errors here. So these are things that cause it. So these are things that we can control. So why we're going over them is because we can control them. I'm not going to go over genetic. You have no control. Okay. So the things that you want to avoid are physical mutagens. So physical mutagens means that you go in and actually, these things actually tear the DNA apart. Okay. They can cause point, frame shift, and chromosomal errors. They're basically going in and basically ripping the DNA apart. And if the DNA is all ripped up, it can't replicate properly. So the most common ones are things like X-rays. So if you become an X-ray tech, okay, you can't be in the room. That's why when they do your X-rays, the person actually leaves the room and then does the X-ray and then comes back in because they can't stand there. You're okay because you're only getting 1 X-ray, but you're not going to be able to get 10 X-rays in a day. They would never do that. They would give you cancer if they did that. You can only get a certain number of X-rays over a certain time period. And X-ray techs who are doing X-rays all the time, They don't want to expose themselves to all that, so that's why they are always leaving the room. Okay, it's also why you put that lead jacket on, or they'll cover certain areas of your body that are not being x-rayed, because they don't want to expose your heart to an x-ray if they're looking at your shoulder or something like that. UV rays, the sun, same kind of thing as x-rays. It's basically some photons. Here's, if you're from a chem class, photons are going to come in, and the photons basically hit the DNA and then start warping it. Okay, so UV radiation causes a reaction between actually C and T. So that connection, okay, and can cause skin cancer known as melanoma. Okay, so you really want to avoid bad sunburns. Okay. Besides physical, there's other things like chemicals that we take in. They're called chemical mutagens. So there's certain chemicals that if you ingest or you breathe in or you get them into your bloodstream somehow, they can cause mutations. So they mostly cause substitutions and frame shifts. The most common are nitrates, gasoline fumes. That's why you can't have gasoline open indoors. Okay, and why it has to be fumigated. Pure carbon is really, really bad for you as well. Okay, and that's what charcoal is, or if you eat a burnt hot dog or burnt toast. There's a whole bunch in cigarette smoke. It's like it just doesn't even make sense on how many different chemicals cause cancer in cigarette smoke. So really, really bad for you to smoke cigarettes. Chemical mutagens that cause cancer are known as carcinogens. That's what the term carcinogen means. Basically, it means a chemical that causes cancer. Okay? There's a huge list of them. I'm not going to go through them. I usually do this in grade 11. I mean, when people say there's a lot of things that cause cancer, it is true because there's a lot of different carcinogens. All right, the last one that we're going to talk about, the last one that you can actually get a mutation from, is maybe surprising, is biological. A virus can come inside you and actually mutate your DNA. Okay? A bacteria can also do it. So just because you've got a viral or bacterial infection doesn't mean, oh, that's not going to be long-term or there's going to be no cure. Okay? A virus can go inside of you and cause cancer. Okay? One common example of it is it's called HPV virus. Okay? And all the females in this class, and potentially the males, depending on how old you are, got vaccinated for this when you were in grade 7 or 8. Depending, some people got vaccinated in grade 6. And what they did was they vaccinated you for this. They gave you a vaccine so that you don't get this virus. And the reason why they did it primarily with females was because the virus attacks your cervix. Males don't have cervix, so it doesn't really affect them. But the virus attacks your cervix, and when it attacks your cervix, it starts attacking the cells in your cervix, in the DNA in those cells, and can lead to cervical cancer. Okay? So they basically give you a vaccine so you don't get this virus, and then you can't get, or reduces your chances of cervical cancer. They give it to males now because it's actually an STD. So males, even though they're not affected by it, they can give it to females. So males are now also being vaccinated for it. Okay, just as a more preventative measure. Another type is this H. pylori bacteria, which can cause ulcers. Okay, and an ulcer is that your stomach cells were mutated and they now don't produce mucus anymore. If those cells don't be able to produce the protein mucus, then they're not protected by stomach acid, and then you get a burned hole in your stomach. Okay, so there is bacteria and viruses that can go in and actually start altering your DNA. Okay, the last thing I just kind of want to go through is a couple different diseases where we kind of know exactly the reason for it. And one of them is Tay-Sachs. Okay, so this is an example of Tay-Sachs. So these are the signs and symptoms of Tay-Sachs. Okay, you're going to have a series of different symptoms like loss of learned skills, loss of smile, so the babies don't smile, which is a telltale sign, blindness, deafness, paralysis. And again, all of these are the same because they all have the same genetic deficiency. Okay? And since they all have the exact same genetic deficiency, they all have the same code that's wrong, they all have the same proteins that are wrong, they're all going to have the same effect from not having those proper proteins. Dementia, unable to swallow, muscle atrophy. So when you pick up a child with Tay-Sachs, they lay limp like this, which is very, very odd for children. Usually they spring up. Okay? And then a cherry red spot in the back of their eyes. Because without this protein, you are unable to do all of these different things that we're listing here. And it leads to a telltale sign. Now, what causes this? Okay, what they've figured out that it's actually a point mutation. Okay, right in this gene called a hexa gene. So the allele is just a version of the gene. Just replace allele with gene. So this hexa is the gene, and it makes a protein that's vital. Okay, so you need to make this very, very important protein. And what has happened in someone in Tay-Sachs is that they've got an altered reading frame. Okay, is they got an insertion. Okay, it should be TCC. Okay, and we got TCT. Okay, and if you notice, when that happens, now we get this downstreaming effect of all of these amino acids are wrong. Okay, this is the amino acid sequence it should be. It should be serine, then tyrosine, then glycine, then proline, and then aspirin. Okay, that's the sequence that should happen. But because we got a frame shift, now we have an amino acid like this. Okay, uline, leucine, cysteine, proline. Okay, and because they lack this, this protein is very, very important in the body. Variety of symptoms. Okay, another one is muscle dystrophy or Huntington's. Okay, so they know exactly where the DNA sequence. If you look at the DNA sequence, they know exactly where this is. Now, knowing exactly where this is why when babies are being born, mothers have the option of doing genetic testing. So if you have a history of Huntington's disease, okay, you can get your baby prior to being born to be checked, and they can tell you, based on the DNA profile of the child, it does have Huntington's or it does have muscle dystrophy. Okay, and what I already went through. And then again, just kind of talking about chromosomal errors. So if you have an entire extra chromosome, like here in Edwards syndrome, where the child is born shortly after, dies shortly after death, or again the amazing Down syndrome individuals that even though they have a lot of extra information, okay, the Y Down syndrome, one of the reasons is their extra pieces of information is on a very small chromosome, okay, where something like on Edwards, it's an extra piece of information on a larger chromosome. If you even go to chromosome 2 or chromosome 1, these are all basically miscarriages. They're not even being able to be born because you have way too many genes. But if you get down here and like 21, there's not a lot of genes on that chromosome, which is one of the reasons why Down syndrome people are so capable. Okay? I hope this helps with mutations.